Okay, you guys, this is Unit 2, Segment 3. This is the last segment for this unit. The key vocabulary words are enzymes, catalyst, activation energy, lactase, and Benedict's test. Flashcards would be great for this. Starting with catalyst. Catalyst is a general word. You use it in chemistry to describe any substance that will speed up a chemical reaction. Now, in biology, we are looking at biological kinds of catalysts, and we call those enzymes. They're a type of protein. So I wanted to give you an analogy. You're going to need to know how this looks graphically, so I wanted to give you it as an analogy. So we have energy going up this axis, okay, the amount of energy going up, and then time is increasing as we go to the right. Now looking at the two lines, we have one going very high and another one not going very high. So when we talk about a chemical reaction, we have reactants going in, there they are, and products coming out. So when the reactants have to travel up lots of energy, okay, to travel in order to make the products, that means there's a high what's called activation energy. And this would happen in your body just fine, but it will take a lot longer because it requires more energy, and it might have to happen even at a higher temperature. But without a catalyst, that's without a catalyst. This is what would happen naturally. Now with the catalyst though, you can see this man just has to merely just push it with his hands. Much less energy required for that when there's a catalyst. So my friends, catalysts don't make the reaction happen. Catalysts help the reaction to happen. So by any chemical reaction, the two reactions that we talked about so far have been dehydration synthesis and hydrolysis. So when we're talking about making and breaking polymers, there are catalysts in our bodies that will help speed up the hydrolysis and the dehydration synthesis. So you can see without the catalyst, it's a lot harder for the reaction to happen. It takes a lot longer and it requires more energy. Now, the biological catalyst is called an enzyme. You have enzymes in your body. So we won't, if you're in chemistry, you're not going to necessarily talk enzymes. But when you're in biology, now we're looking at biological catalysts, things that are found in your body. And these are proteins, so they are an actual macro molecule. And they will speed up the reaction that's happening, the reactions that are happening in your body. The type of biological catalyst. So now thinking about the analogy graph that you saw on the last slide, you have, this is a real graph. So you have energy going up as you go up this axis, and the reaction time is progressing as you go to the right. You start out with your reactants, you go through the reaction, and you end with your products. How much energy, okay, is required before that reaction happens is called activation energy. And tell me, this is without the enzyme, this is with the enzyme. Which one requires a higher activation energy? The answer is the one without the enzyme. That's because that goes much higher. So without the enzyme, your reactions will happen, but they'll happen much slower and require more energy. With the enzyme, it's much faster, the reactions are much faster, and they happen with less energy. And the same products are being produced. So enzymes are your friends. They will allow these chemical reactions like hydrolysis and dehydration synthesis to actually happen at a pretty good rate. You are going to be asked to describe the lock and model key, okay, on an assessment. So enzymes, there's some vocabulary words. There's enzyme, substrate, active site, and the enzyme substrate complex. There's a lot of vocabulary words in here. I need you to look at these images as I describe what's happening, and then I'm going to show a video too. So here, the two reactions that we mentioned before are hydrolysis. Okay, this is where we're breaking polymers, and then we have dehydration synthesis where we're making polymers. So here we have a substrate coming in, which you could recognize maybe as a polymer, okay, as a polysaccharide or as a protein or a lipid, any kind of polymer that comes in, and it perfectly matches the enzyme shape. See how it's perfect like a lock and key. So the key would be the substrate, the lock would be the enzyme. It comes in, clicks in, and because of this perfect fit, it's very specific to that reaction and that particular substrate. No other substrates will fit into it, just that one. And it pulls this in, and because of this enzyme substrate complex, it's able to allow the reaction to then happen to break them apart. So what would these, if this is the polymer, what are these? Answer is monomers. These are the monomers. So we could be talking about any of the macromolecules, fats, proteins, nucleic acids, or carbohydrates, and this is how the monomers form. This is when we take in food in the polymer form. That's our food. We break it apart so that we can remake it with dehydration synthesis, so that we can actually make the products that our body needs. So the enzyme lock and key model can work for hydrolysis, and it can also work for dehydration synthesis. In this bottom example, same kind of thing. Here's our enzyme. Here's our two substrates. What are these called, you guys? Polymers or monomers? What do you think? Answer is monomers. The monomers are perfectly fit for the specific enzyme. They come in. The enzyme allows for the reaction to happen much faster because it's perfectly fit for them. It's like, come here, guys. Come on in. I'll wrap my arms around you. And then because I'm doing this, you're going to react so much faster. You're going to bond. A bond is going to form between that. And what do you think this product is called? In biology, it's called a polymer. So then you can be breaking up your food and then remaking the macromolecules that your body needs. It can be DNA, it can be proteins for your muscles or your bones, it can be lipids that you're going to store your energy with. I mean, it can be any of those things that we've been talking about. So lock and key model, try to write out or describe these images because you're going to be asked to do that on an assessment. Activation energy I've already mentioned in the other slides, but that is going to be the difference between, activation energy is the energy needed to start a reaction. So when you have an enzyme, notice the difference between the height that this one gets versus the height without the enzyme that it requires. So when there's no enzyme available, the reaction will happen, but it's going to require a lot more energy, a bigger climb. Remember that guy pushing that wheelbarrow? He has to push it so much higher, it's so much more energy. And so then the reaction will happen and the products will be made. I like this illustration because it shows going in, we have reactants, then the reaction happens, the enzyme substrate reaction happens, and then we have our end product. And that's called, and the energy required is called activation energy. I think I'm going to pause this for right now, and I want to show you a quick animation. I'm going to go ahead and just play this.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site.
Come on, guys, join me.
The enzyme-fit substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates.
Now it's reacting.
These stresses
forming the bond
link between the two substrates, leading to the formation of a different molecule.
Polymer.
As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
Basically, he's saying, here we've watched a polymer be formed, you guys. So dehydration synthesis, formed the polymer, and then it can also work the other way. The enzymes can take this polymer and then break it also, and it would be a different enzyme to do that. So you have enzymes that your body, your DNA is coded to make. This is a protein right here, and its job, its special function is to act as an enzyme to make this product, to allow for that product to be made. Don't get me wrong, enzymes are not making the chemical reaction. They're allowing it to happen at a faster rate, okay? So it comes out the same way it went in, but what happens is this product is now made where it wasn't before. Let me go back to the presentation. So there's a couple of enzymes. There's one in particular that you guys need to know, and that is lactase. So this is an example of an enzyme that your body makes. Some of us don't make very much of it, and so we would be called lactose intolerant. But lactase, A-S-E, if it ends in ACE, it's an enzyme. If it ends in OSE, O-S-E, it's a sugar. So anyways, lactase is named because it's a particular protein enzyme that helps to break down lactose. So the enzymes are actually named for the reaction that they're allowing to happen. So lactase helps to break down lactose. And here is a image showing the molecular structure. So if you look, you can almost make out a monomer. Right there's a monomer. Right there's another monomer. Look at their joined. So this, my friends, is a polymer. And it happens to be because there's two monomers together, and this is a carbohydrate, lactose, think of glucose, fructose, all those oses were carbohydrates. We call this a disaccharide because there's two. You guys learned about polysaccharide, more than one, but there's one monomer connected to another monomer, so now we have a chain, we have our necklace, and that would be like a polymer, but because there's specifically two, it's a disaccharide. And this is lactose, okay? That is found in dairy products, milks, cheeses, ice cream. And if you've ever heard anybody say, gosh, I'm lactose intolerant, that means that their body is not, their genetic, their DNA is not coding for their cells to actually make lactase for them. So they can't break this up. They can't digest it, and it causes digestion problems. But so they may take lactase, a pill, that actually has the enzyme that allows your body to then break it up into the simple sugars. Notice how we have two separate sugars. These are monomers. So you have polymer broken into monomers. What is that process called? Hydrolysis. You know it, hydrolysis. Lactase helps that happen, okay? It's an enzyme that helps that. It says, come on in. And then it'll take that in and it'll break it apart. It'll allow that reaction to happen. It's going to help it happen. Benedict's test is one that you did in the lab. It is an indicator test for simple sugars. So if we were to throw in lactase into milk, it would break the lactose into the glucose and the simple sugars, and then we would use the Benedict's test. And if you recall, it was blue, and then you put it in the hot bath, and it turned, if it was positive, it turned orange, sometimes even red. That will show us that it is successfully, that lactase is successfully breaking the polysaccharide into the monosaccharides. So I'm going to show you some more with that too. I wanted to just walk one more time through. I have about a minute and a half of time. So what happens when the enzyme is present? Here again, you're showing two lines. We've got reaction time happening across the bottom, potential energy going up the side. With no enzyme whatsoever, it's going to require more energy. That guy pushing that wheelbarrow is going to have to go higher up before the reaction can actually produce the products. If there is an enzyme, look at the difference. It's much lower, okay? I also want, before I close this out, this is, by the way, this is activation energy, the difference between the two, but before I close this out, I really want you to know that enzymes sometimes are they are temperature sensitive. Some enzymes will only function in certain temperature ranges. If they go beyond that, then they won't function anymore. It's actually denaturing the protein when that happens. So for example, when you cook meat, you are denaturing the protein. You're actually changing the shape of it, and it won't break down. I can't go back to raw meat after it's cooked. So enzymes are particular. They will function. They will peak function at certain temperatures, at certain pHs, and everything. Those are kind of there are parameters to which they will work. And I wanted you to be aware of that. And that is it, you guys. That is all.